Welcome back, friends. In this video tutorial we'll be learning about another alcoholic beverage preparation, and that is beer. Now in these videos about the different alcoholic beverages, we are majorly focusing on the biotechnology and industrial microbiological perspective of how the microorganisms are acting to ferment the sugar into other alcoholic compounds like wine, beer, whiskey, and so on. Excuse me. Now, so let's begin. So let me take a color first. Let's take the floating tool and a color. Okay. So beer is world's oldest and possibly dating back to 6000 BC. So you can see how old this alcoholic beverages actually is, and this is very famous, right? And it is the third most popular drink after water and tea. Okay, and it is actually produced via the process of fermentation, and the fermentation process to produce beer is simply called as brewing. It is called brewing. So when you talk about brewing, we are talking about the production of beer from the starchy material or polysaccharide containing material. Okay, now usually the beer is produced mainly from cereal grains or the fermenting cereal grains. And the most common of these cereal grains that are used for the production of beer is malted barley, right? So barley is taken from the ground, they are harvested, and we take the barley and we allow this barley to be malted. So malt is being produced from the barley, the process called malting. After the malting is done, uh, we take that malt and we use that malt as the starch material, as the source of the starch material, to finally convert the starch material into what we know as the alcohol, right? So here it is, this is the malt, and barley is taken first and then we convert them into the malt using simple germination process. Then we take the malt and we convert them into, we produce a mash of the malt by mixing water to it, and we then finally add another important ingredient which is called hops, right? In beer making actually there are four major ingredients. One is this malts, second thing is water, obviously water is required, third thing is yeast, and fourth thing is hops, right? Actually I put third is hop and fourth is the yeast, which is the organism which will convert excuse me, which will convert this sugar into beer or alcohol, right? So hops is the important ingredient because it is providing the characteristic bitterness to this beer, right? And it is responsible for the flavor. So this is a picture of hops. As you can see, it's again a plant material. We can dry the hop, we can put the dry hops there, and finally what we get, we get the beer. And we must require yeast here. In the picture we haven't put yeast here, but yeast is also required for the process. So now if we look the type of beer, what we can see, there are two types of beer actually. One is ale beer, another one is the lager beer. Ale beer is the top fermented beer and lager beer is the bottom fermented beer. Now in the ale beer, it can be divided into three different types: pale, middle or brown, and stout, right? In the pale beer, it is definitely the pale color. As you can see, this is the most common one, like that. And second thing is a middle or brown beer is also there, and another one is a stout or porter, which is a black and dark colored beer. And all of this beer, pale beer, brown beer, and stout beer, all of them are the part of ale. Ale means obviously the fermentation of the beer or the fermentation of this malt is actually done in the top region. So if this is the chamber of fermentation and it is filled with malt and water, the fermentation is going on, or it is carried out by all those individual like yeast in the top region. So top region fermentation is done in this case. Okay, but there is another type of beer that is called a lager beer. Now the lager beer are bottom fermented beer. That means in this kind of beer, in the lager beer, fermentation usually carried out in this bottom region. So in case of lager, we are having fermentation at this bottom region, and in case of uh, ale, we are having fermentation at the top region. Okay. Now lager beer are also of two different types. One is pale, which is this color, and another one is a dark, which is again dark black color. But there is no brownish color beer in case of the lager type, right? And the pale beer is also divided like Pilsner and Dortmunder. These are the different examples. In the dark beer, we are having Bock and Munchener. There are different names and examples. I don't encourage you to memorize, but you must know that there are the different color variation like pale coloration, dark and brown. both this kind. Usually the browns are ale, and those things can be distributed. Now the production of beer. So in this picture, as you can see, the whole stage of beer preparation is illustrated pretty clearly. I'll be talking just overview of each of the stages. So here we get the barley. Once we get the barley, the first job is the malting of the barley. That means the germination of barley, because the germinated part of the barley is called as malts, right? So for this malting, what we need to do, we need to add some water, because they require moisture for the germination, as well as they require some internal hormonal activity like the gibberellin activity inside the barley to finally convert them into the excuse me, malts. So this particular process is called steeping. In the steeping process, they are receiving moisture from outside, as they're receiving in a bed where it is getting all the necessary moisture and everything required, and obviously they require some optimum temperature. They start to produce or germinate. They start to germinate. Here you can see germination is going on, and things are coming out like that. Okay, so germination is going on in this case, and then finally after the germination is done, when they do not require moisture anymore, we need to take out the moisture out of this place. For taking out that moisture, we need to dry them, right? Now that part is called the kilning or kiln drying process, right? So once the moisture content is utilized, we need to kiln dry those malts, and after the kiln drying, the step is called the milling, because we need to make powder from those malts. Right now the malt are of several different varieties. It can be of this yellowish color malt, it can be of dark color malt, so light or dark, both the type. Some beer are prepared with the dark colored malt, they give rise to dark colored beer, and some are prepared with light colored malts. So here we can see the roller mill. What we add all the germinated what we can say barley or malt there, and we produce a grist of it. Once we produce the grist, we are having a mixer or mash mixer. In this mash mixer, once we get all the grist, which is the powder portion of these malts, are added, and we add the another important ingredient or second important ingredient, which is water. We have already talked about one important ingredient, which is barley or malt. This is the second important ingredient. Water is added here in the mash mixer. Now in this case, what we added, this grist is mixed with water thoroughly, and what it is producing, it produces what is called a mash. Now mash means we are having malt with water mixture, right? So once we get this done, we take that and we add it into another chamber. It is called tun or lauter tun. In the lauter tun, you can see this color, it is made up with copper. This is very important. To produce a quality beer, the lauter tun and rest of the process where the fermentation will go on must be made with copper, because copper the presence of copper is actually sometimes help. There are some different theories, but usually some scientists are telling that this copper can act as an important enzyme cofactor during the process of fermentation, right? That's why copper is important. So they impart some of the they can help to achieve the fermentation pretty quick, right? So once they start to add everything in this lauter tun for several hours or for some time, then what they do, they transfer it into what we call a kettle. It is also made up with copper. It is a giant kettle, as you can see, and it is having a typical characteristic or having a kind of chimney like structure, though the top region is sealed. It's not open like normal chimneys, it is sealed, and this is called the kettle or lauter kettle. And this kettle, what we can see, this is the region where everything will be mixed properly, right? So they add it here, and slightly they will heat this whole mixture. So they add everything. So once they mix, this mash is taken from lauter tun into the kettle, it will be called wort. So these are the terminologies: mash, wort. So everything is the name after making the preparation. So what is provided, and once the wort is provided into the kettle, they add hops. Hops is the third most important ingredients in this case. So they will put hops there into the wort, and after the putting hops in the wort, the actual brewing process begins. And in this case, you can see this kettle, how large these kettles are, and it is respective to this person. This is very large. So they put them everything like that, and then after some time they will take it, and what they do, they will, so they will heat this kettle for some while. So as a result of this heating, all of the ingredients that are present in malt are coming out to the malt water mixture into the wort, right? So into the wort, the mixture, every flavor is kind of mixing, because flavor from malt is kind of coming out, flavor from hops are kind of coming out, so everything is come out and they are mixed with the water. So once they're mixed with the water, so mixture is containing every little bit of that typical beer flavor. Now once everything is prepared like that, so the mixture is heated. Now we need to cool the mixture, so we need to transfer that mixture into a plate heat exchanger. So here it is, the cooling chamber or plate heat exchanger, where everything is put and it will be transferred from one after another plate, so that the heat is kind of dispersed to the plate and the mixture is becoming cool. So once the mixture is now cool, now it is being ready and subjected to the actual fermentation. Now I remind you, in any kind of alcoholic beverage preparation like beer or wine and all this thing, before the fermentation should be carried out in a mild temperature, not in the high temperature. So in all this case, if you see that, if the mixture is heated or everything is done, but still after everything, before the fermentation, you need to reduce the temperature of your mixture. So you reduce the temperature and put it into the fermenter. So once you put it into the fermenter, now is the time to add the last and the most important ingredients, that is yeast, in this particular stage. So yeast is now added. So now as we add yeast into the fermentation mixture or fermentation chamber onto the wort, the yeast will start to ferment, right? Now during this process, what it produces is called the green beer. The green beer means immature beer, just the start of the fermentation, slightly. So here in this first part, we're having lots of sugar, and then we add yeast, so we are having very low alcohol, because there is no alcohol there. Now as this beer start to ferment, what it produces, green beer. In the green beer, slight alcohol concentration, slight alcohol is start to form, and sugars are start to degraded. Now after certain time, there is a maturation tank where the fermentation is going on for full flog, and it is going on rapidly. So as the fermentation is going on pretty fast, all the sugars that are present start to convert into alcohol, right? So sugar starts to convert into alcohol by the presence of this ingredient yeast, right? Okay, so yeast is converting it into the alcohol, so less sugar is left, more alcohol is there. Now once the process of fermentation is conducted, we need to transfer that mixture which is filled with alcohol, almost 6 to 8% of that, not 6 to 8, actually it's beer, so it will be less. It will be approximately 4 to 6% of the alcohol that is being generated. We transfer it into the filtration unit. The filtration unit can be of different type, clarifiers or filter and any kind. So using that filtration unit, we can filter out our beer, and once we get this filtering out of the beer, we bottle that into the different bottling. So bottling is done after the filtration. After the bottling, it's the time for a kind of tunnel pasteurization. Now why we require this pasteurization? Because very sorry, I'm very sick right now anyways. So here anyways, we get this beer and we bottle that beer. So once we produce this bottling, now I remind you, in all these stages like lauter tun, kettle, a separator of the liquid from this whirlpool separator, which is the separator agent for separating the liquid part from the solid parts. So in all these things, all the chambers must be fermented, and all this chamber must be sterilized prior to the process of beer preparation. So it is required. After all this thing, we need to go through another round of what you can say sanitization, or another round of disinfection. Otherwise it will be very much contaminated with some other microorganisms. We don't want those contaminations. So for that reason, one final step of pasteurization is carried out, and this is called the tunnel pasteurization. So once every bottle is ready, it is capped, now it is moved through a kind of conveyor belt, and through the pasteurization is going on there. After the pasteurization, via the UV irradiation or something is going on, after that, if there is an organism left in this bottled beer, they will die and they will lose their viability. And after that, what we take it, and then we make a leveling of the bottle, and we finally did it. And Remember, in this particular step, after the kettle, when you take it in the whirlpool separator, there are two important things are generated. One is liquid, another one is solid and liquid. So the liquid materials that is generated is simply turned into the fermenter tank, and the rest of the process will be going on like that. But for the solid material, it will be transferred and to the flash pasteurization, and at this particular stage so they will be taken as a cattle feed. Okay, now some of the beer are directly bottled after this filtration step and pasteurized and ready to be provided to the distributors, but some of those beer is then taken into the flash pasteurization, then they are placed inside a keg. Now this keg are also being prepared, not like bottles, but keg are long and larger chambers where the beer is this keg is acting as a reservoir for beer, and these kegs are delivered to bars where lot of beer supply is required, less than the different bottles. So we provide the keg to those for that purposes. Okay. Okay, so let's move on. Let me take a color here also. It's okay. Okay. Okay, now let's talk about the brewing in little bit more detail. Now the process of making beer is simply called as brewing, right? So I've already talked about that. Okay, and once we get everything in the water, like the extract from the hops and the extract from the malt is called a wort. So that watery part containing all the important sugary materials to be fermented is called the wort. Now, the first important step is the malting of the barley. Now, the barley is selected in use for the malting industry and must meet special quality specifications. And actually, the accepted malting barley varieties have to modify to get the better quality beer from time to time. So the first step for the malting. So before the malting, you need to know about a little bit of a structure of the barley. So if this is the barley, what we can see in this case, this is the outer membrane. It is called the husk of the barley, and in each of different stages of this barley contains specific type of nutrients which are coming out during the wort preparation. So husk is having a type of ingredients, embryo is having a type of ingredient, then endosperm is having the rest of the starch material. So most of the starch material is coming from this endosperm region, and aleuron is having its own. So here in this case, in the mature barley, the embryo and the endosperm are the most important stages for the preparation of beer, right? Now the store for carbohydrate is this beer, so this is having carbohydrate and this is also having carbohydrate. Rest of the part are having less amount of carbohydrates, right? And usually this is the starch layer, this layer is the starch layer, this whole endosperm layer is the starch layer, and surrounding this layer we are having aleuron. This is also a living, this is a kind of non starch but living cell wall layer. Okay, let's talk about now vitality of those what you can say malts or barley. When stored at very low temperature and low moisture content, can be vital for centuries. That is very important importance about this barley, because they can be viable for centuries if we store them in the right temperature, like 12 degrees Celsius temperature and moisture of below 12%. Higher moisture content can spoil those barley because they can have fungal contamination. The moisture above 14%. If you store barley at moisture above 14% for several days, it can cause several fungal contamination, right? Aspergillus fusarium like of contamination. Okay, and what they produce in this particular stage is some secondary metabolite, and those secondary metabolites produced by fungi can cause kind of situation called gushing, as you can see in this picture, simply a kind of foamy beer like this. Okay, now these are the different stages of malting. Actually, as you can see, these are the different stages. So here it begins with that. So these are the stages. In this upper region, it is stained with dye, and this is without the stain. But these are the different stages from different days. Actually, this is not days, it will be a days. In day 0, day 1, first day, second day, third day. From the third day on, it is kind of germination is kind of coming out. You can see it, visualize it. Day 4, day 5, and fully matured malt is we get fully matured malt. Now this is very important to choose the particular stage of harvesting, to choose the particular stage for the preparation of beer. You cannot take this first day malt for your beer preparation. That is completely wrong. You cannot take 6 or 7 day, because it will provide some long trees, right? So you need to take it from the day 4 and 5, like that. So this is a sequential events of malting from barley to malt. So storage is a moisture from 12 to 14%, but not more than 14%. Temperature should be less than 12 degrees Celsius. It's actually 10 to 12 degrees Celsius. 12 degrees Celsius works most fine. That's why we provide that. Steeping is the process for malting. It's a moisture of 45%. It is required for the steeping. That means this is not about the moisture for those barley. This is the moisture of your environment actually. For the germination purpose, you require the moisture of 45%. Now in this particular stage, what we do actually produce a bed. In the bed, we kind of produce a moist bed as kind of moist chamber. In this moist bed and a moist chamber, we just put all those materials there. So let me put all those, say these are this green color thing are all those barleys. We put all those barleys and we allow them for 3 to 5 days. Now actually the germination time is 5 days. You can see in this previous picture, almost in 4 to 5 days we get a proper shaped properly produced malt ready to be transferred for the beer preparation, right? Okay, and the temperature is 12 to 16 degrees Celsius for the germination. So actually the whole process of malting is divided into 3 different stages. Steeping, first stage is steeping, second stage is germination, and third stage is kiln drying. First stage is the steeping, which is we need to provide a higher moisture content uh, for this barley to relieve its actual stage and to be moved towards the germination stage. Now during the germination process, we require very less amount of moisture, like 12 to 14%, and the temperature must be 12 to 16 degrees Celsius temperature. After the germination is done, we don't require moisture anymore, so we need to dry the situation. We need to dry those malts, so we need to dry it before subjecting it for the beer preparation. So the drying is called kiln drying, and the temperature in this case is almost 85 degrees Celsius temperature. The moisture is up to 4 and less than 4% of moisture, right? So as this process is done, we need to cut those rootlets out. As you can see here, stemlets and rootlets are coming out, so we need to cut these rootlets from this region, but we need to take the malt, because during the germination, the malt is providing all the necessary nutrients that is required for the perfect beer preparation. In the malting, what we can say, you can see the uptake of water is in the steeping vessel is really, really important. So we just put all those barley into the steeping water. So we can see the moisture content is 45 to 50%. Now followed by the aeration, and until the water content grain reaches 42 to 48%. Now the grains are then transferred to the malting beads. So here you can see the malting beads are coming out. Here is the malting beads, and here they allowed for the germination for 5 days. Once everything is produced, we call it the green malt, and after the 5 days we need to subject them for the clean drying. So in this case, these are the 3 different stages. This A stage is the steeping. So you can see in the steeping, we need to dip all those barley into the water. The second stage here is termed as germination. Actual germination is taking place here, and rootlets are coming out. After that, we need to put them in the hot environment. Here it is a heater coil. So it's a schematic presentation like that. So actually in large productions, we don't get this kind of heater like structures. Kiln drying is done in different ways. We'll be seeing the picture later. So in this particular stage, when they are heated and they are ready to ferment, it is called the green malt. In this particular stage, this is called the kiln drying. This is called kiln drying. Okay, so these are the 3 different stages. Now the important action of hydrolase and different enzymes during these germination stages for barley to malt. This is very, very important. First is a rapid and uniform germination. Second is the synthesis of hydrolytic enzymes, which is required, and these hydrolytic enzymes are generally produced in aleuron. Remember, we have talked about aleuron, which is a living tissue there surrounding that endosperm, right? And the third thing is the degradation of the endosperm cell wall, which is required for the starch to come out so that the fermentation can be carried out by yeast, right? So it is described as a modification of this malt, right? The final stage, or the degradation of endosperm cell wall, and the release of starch into outside. Now for whole of this process to be tightly regulated, the hormone gibberellin plays the most important role in this case. Okay, presence of gibberellin is very very important. Now here it is. So here we can see this is a malt, and enzymes can come out from this allurone, which is here like that. So as the enzymes are coming out, gibberellin is the most key player because it is regulating everything. So once the enzymes are coming out, starch is being degraded into maltose. Now the maltose will be further degraded into glucose. What are the enzymes for doing all these things? Remember, all of these enzymes are called hydrolytic enzymes. So hydrolytic enzymes will work onto it, and they'll bring or they'll break down the starch into maltose, then break down the maltose further into glucose, and the glucose can be taken by this embryo pretty easily, and embryo start to grow. That is the actual funda of the malting. It's a simple germination process, and for that they require water. So that's the reason for steeping, 45% of moisture. After that, when the fermentation is carried out, the moisture level should be 12 to 14%. Then we need to reduce it into 4% in kiln drying. Okay, so every different stage, every single stage is tightly regulated during this process, right? So that we get the perfectly quality and perfectly what we can say, we get the perfect form of malt for our preparation of beer. So here we can see the steeping and germination stages differently. Now this is the step for steeping, where we dip them in the water. So after dipping them in water, approximately the percentage of moisture 45 to 48%, then we put them into the ground like that. Then we put them into the ground, and here we can see the germination is going on. After the germination is going on, what we need to do, we produce the green malt. So once we produce the green malt after the germination, we need to allow them to dry for some days, and that is called the kiln drying. Remember, here it is a process of kiln drying. So it's a large surface area containing region where you put all those malts, all those germinated malts there. So they will come, and at this level the actual idea is to low down the moisture content inside the malt. So we bring it less than 4%, and approximately pretty close to 4% in this particular time. So after doing all these things, the malt is required to be milled and will be mixed with water, right? That's the actual process. So for that reason we take this malt. So again, schematic representation. So we take the malts, we are having a kind of what you can chamber, which will kind of mash and make the powder of the malts like that. So once we make the powder of the malts, it will be taken, and so let me light, it is called the malt powder, and we add water to it. Once we add water to it and you start heating them, so heat is applied in this region, so they kind of produce a kind of thing, it is called the mash. Remember, we have already talked about that, and the process is termed as mashing. Once the mashing is done, at the end of the mashing, the soluble substances are and the residual solid particles are separated by a kind of whirlpool filtration unit. Remember, we've already talked about that. We've already seen the picture previously, right? So here, after the filtration, what we get, two important things. One is the solid material, another one is the liquid material. The liquid material here is called the wort or sweet wort. Why it is called the sweet wort? The terminology for that is wort, and it is called sweet because it is still consisting of some sugar. Because remember, fermentation is not done yet. So all of these stages are the processing stages before fermentation, and that is very very important, because fermentation process is actually taken for 1 to 2 weeks like that, not very long, but the processing is very very important. If the processing is not done properly, fermentation will not work properly. So that's why processing is very very important. So once as we are having sugar with this wort, we call them the sweet wort, right? So we get the sweet wort, and what we get, the solid materials, we call them spent grains, because they are wasted, they are already spent. So we call them spent grains, the solids which are taken for the cattle feeds and for the fertilizers. As a result, they can produce fertilizers, and the sweet worts are taken, and the next step is to add the hops into the place, and we heat them in the boiling temperature of greater than 1 hour in this particular chamber. This is called the kettle. Remember, boiling kettle, and this kettle should be made up with copper, right? So we put them in the kettle and we add hops here. So we are adding hops. You can see the picture of hops are added. We are heating them for 1 hour like that, or 1 to 2 hours, and this is the actual step of this boiling in the copper. During this stage, actually, the ingredients are coming out from the hops. The ingredients, nutrients and flavors or aromas are coming out from the hops, as well as they are coming out from the malts, and the kind of mixture is generated. Once everything is generated like this, so we require remember, the mashing step is very very important. So we haven't talked about mashing in detail. So in this picture we talked about all the stages little bit or schematically. Now let's talk about mashing in detail, then we'll be talking about addition of hops and all this. Okay, so mashings are normally preferred at the pH of 5.5. So it's a kind of slight acidic pH, because this is the pH at which the most malt derived enzymes exhibit the proper activity. Okay, and this process of mashing is nothing but the enzymatic activity. Some more enzymatic reactions are going on. As we can see, we are gradually increasing the temperature from 38 degrees Celsius to 77, and finally to 85 degrees Celsius temperature in this particular stage. Now we're gradually increasing the temperature, right? So just increase little bit and stabilize for some time, increasing and stabilize, so like that. Okay, now why we are doing so? Because there are certain enzymes present in this malt that are acting at 38 degrees Celsius first. So we allow some time for those enzymes to work perfectly at this 38 degrees Celsius temperature. Once those enzymes are doing their job, they have done the functioning their job, we just slightly increase the temperature to 52, so that those enzymes present in the malt, they act properly on 52 degrees Celsius temperature, can act now. Once they have done their work, then we increase it to 68, so that another round of enzymes which are having the optimum temperature of 68 degrees Celsius can function properly. So the actual idea is our malt is consisting of many enzymes actually. So if I draw the enzymes, let's say this is an enzyme, this is another enzyme. So let me change the color for the enzymes. These are another enzymes. These are red, say these are green. So there are a lot of varieties of enzymes like that. And among these enzymes, what we know is that, let's say this black enzyme works properly in 38. So if we provide the 38 degrees Celsius temperature, they will work fine, they will done their job. And then it remains this red and blue, because red and green, right? Because red and green won't work in this 38 degrees Celsius temperature. So increase the temperature 52 degrees Celsius. Now red are active, so red will do their job. After that, this green will act as 38, and there are other enzymes which will act on 77. So there are enzymes, sequential enzymes, which are acting in different temperature. That's why we are gradually increasing the temperature. Okay, and the enzymes that are required for this mashing stage, for the production, for the degradation of cell wall, then protein degradation, then start gelatinization, and the saccharification processes, we require enzymes like 1C or 1,4 beta glucanase, xylanase, and cell wall degradation for the cell wall degradation, endopeptidase for the protein degradation, and amylase for the starch degradation. So there are enzymes which require for the degradation of all of these materials. Especially we require this amylase for the starch degradation, because a giant part of this malt is made up of starch. So we don't need to allow, or we don't need to add enzymes from outsource, because the enzymes are already present in malt. We just need to trigger around the different temperature barriers, so that those enzymes work properly. So that's what we do in the mashing stage. After the mashing is done, it's the time for the filtration of the mash. So we take this mash and we put it in the filtration unit. So here it is, a mash filter. It will look something like kind of structure, but here it is a kind of filter. So multiple barriers are there. We add the mashed region. So it is consisting of solid as well as liquid. So we add them into this mixture. So as we are adding them into this filter chamber, the solid are attached and the liquids are coming out, and they are called worts. Remember, they're called worts, and the wort is called sweet wort because sometimes here the sugar is also remaining, because the starch are already being degraded into the simple sugars like glucose, right? So we get sugary material. So in the wort, it's just a little bit sweeter than this mashes, so it's called the sweet wort. So we take this wort, and then is the second stage for wort. So this is the stage for adding hops now. So once we get the wort, so hops are added into the worts, right, or sweet worts after the mashing, right? Now good beer needs a good aroma, and the aroma is and also the typical bitter flavor of this beer is coming from the hops, right? So actually hops is having many different compounds to provide the aroma. It is having a bitter substance almost 19%, oils 0.5%, polyphenols 4%, and proteins 80%. So most, or 20%. So most of the part is made up with proteins and bitter substances. Except for that, it is having a little mineral, polyphenols, and a very very slight oil. Right. So what it is doing here, it converts the amino acids, form of different amino acids, into the isoamino acid structure. So you can see this is the amino acid. It converts the structure into the isoform of that amino acid. So hops are doing all these things, and it is generating typical bitterness. So once hops are added, it's the time for the filtration, fermentation, right? So during the primary fermentation stage, it is called the H, or primary fermentation. The fermentable sugars that are there, we know that wort is consisting of the sugar, that's why it is called sweet wort. So we are having a lot of sugar remaining here, which are predominantly glucose. Now during this process, what we need to add, we need to add yeast from outside. So you'll be adding yeast here. So you add yeast to ferment the sugar. Now yeast start to ferment the sugar into alcohol, and they will produce alcohol here. They'll provide alcohol, right? Along with this alcohol, there is some bitter taste due to the addition of hops, and the aromas are also coming out due to hops, and also the aroma flavors are coming from those malts, right? Now during this process, full fermentation or fully proper mature fermentation is not allowed, because we require some amount of sugar left there. So if you don't, some of this brewing process or brewers require some sugar left there, because they want some little bit sweeter content of the beer, but sometimes they won't require the sugar, so they will ferment totally so that no sugar left there, right? But usually there are some sugar left, and also some yeast also remain there, because some yeast are also remaining there. So we don't kill all the yeast there after the lysis, because after a particular time, when the alcohol concentration is kind of high into this fermentation chamber, uh, due to the alcohol stress, because we know the alcohol can be toxic for yeast, so it is acting as a toxic material, it will start to lyse those yeast cells, right? But still some of those yeast cells still remain there in the suspension, because the cells are responsible for the maturation of the beer in the future times, right? So that's why we leave some of the yeast there for some time. Okay, so now let's talk about the aroma compounds that are coming. Now usually the higher alcohols and their derived acetyl esters are important, adding the important flavors there, important flavor components in the beer. Now it is coming from the alcohol and their derived acetyl esters, right? And it is also coming from hops, right? So they are byproducts of amino acid metabolism in the brewing yeast actually. Okay, amino acids can either be taken up from the wort or synthesized by the yeast itself, right? So that's another important concept, that it can be directly taken from wort or can be synthesized by the yeast, right? One example of the recent research project is the development of brewing yeast with enhanced production of the flavor stabilizer, which is SO2 or sulfur dioxide. Sulfur dioxide can act as flavor stabilizer, as well as it can act as a material to prevent contamination. Because remember, in case of wine preparation, we can see that we enhance those uh, cells to produce SO2 in the fermentation chamber for wine preparation, because this SO2 kind of prevent the other contaminants, like bacterial contaminants there. Okay, so here what we can see, we are having adding all those important ingredients like aspartate and all those amino acid substances, which will be further converted into organic sulfur compounds, which can act as the stabilizer and the flavor compounds there. Okay, now let's talk about another important concept in the beer fermentation and the production of beer, and that is the flocculation. which is required at the end of the fermentation process. Now flocculation is very important because remember, let me explain it to you. Let's say we are having this fermentation chamber, and this is the wort and everything where the fermentation will go on. Yeasts are present there. They are fermenting all those sugars into alcohol, and what they are producing different alcohol. After a certain time, when they provide that stress, when they get the stress due to the alcohol rich environment, in those situations they start to kind of die. So when the nutrient limiting condition arrives for the yeast cell, it is very important that each cell will die and each cell will separate out from, not actually die, but they separate out from this fermentation process into the bottom of the chamber, right? That is very important, and that is called as a flocculation. Now flocculation is required because if let's say this is a fermented yeast, and if the yeast are floating after the fermentation, even after the fermentation, if we bottle them, so inside those bottles what we get these yeasts are floating different parts, so it will be hard for us to distinguish and differentiate between this yeast cell and the beer. So that will be very difficult. We need to add some other clarification processes to take out a pure quality beer, get rid of all those yeasts, right? So what we need to do in this case, we need to apply some process so that those yeast cells are kind of attached to each other, they're kind of adhered to each other, and can kind of settle down into the in the bottle, right? That so that we can easily take them out, and we can easily extract the beer out of this place from the top region, and these settle down at the bottom. So that is called the flocculation, right? Now if we collect those yeast which are settling down at the bottom, we can use them in the future fermentation stages also, right? So that is the process. And how to achieve that, there is a simple theory behind this yeast actually. So if yeasts are fermenting naturally, yeasts are expressing some proteins onto their surface. These are simple called polysaccharides. These are called mannans. Mannans are those polysaccharides. So these are called cell wall mannans or cell wall polysaccharides that are surrounding the yeast cell throughout the region. And there are some other type of protein called flocculins, which are only expressed when the yeast cells are in nutrient deprived conditions. So these flocculins are flow LP or flow 1P. So these are the flop proteins or fall proteins. Now these proteins are only generated or expressed when the nutrient source is kind of depleting in the environment. So when the flocculin is produced, this flocculin is kind of having adherence property to this cell wall mannans. That's very important. So let's say this is a nutrient deprived situations, and rest of the yeast cells are producing these flocculins, and these flocculins kind of go and bind with their own cell wall mannans. So as they are going to go and bind with their own cell wall mannans, they can have a kind of tough interaction, and due to the presence of another important molecule here, calcium, another important ion actually, calcium. So calcium can go and bind, and it can activate these flocculins and mannan receptors. So as a result, they can be eager to bind with their surrounding yeasts. So one yeast can bind with another yeast, and as a result of this flocculin mannan binding, they're kind of adhered to each other, right? So here we can see this flow operon system, flow 1, flow 8, and flow, and all these different things. So they start to express this flocculin proteins. They are going to come and bind with their own surrounding mannans. But now when the gene MNN is expressing in this particular time, what they are expressing, they are producing mannans, and when they are producing these flocculins, they are kind of bind here, and due to the presence of this calcium, it is kind of the region is kind of altered. As you can see here, normally when the mannan is attached to flocculin, the structure or the binding region or the active site of both of these interactions are kind of small. It is not enough to fit onto another mannan of another cell, right? So now as the calcium binds, it alters the structure of this mannan and flocculin. As a result, flocculin is having a larger group now, so it can easily bind with the neighboring cell mannan. So one cell flocculin can bind with another neighboring cell mannan. As you can see in this picture, they're kind of interacting with a flocculin and mannan interaction, and they're kind of what we can say kind of attached to each other, and that's how they form kind of large mass which can be easily settled down into the bottom of that fermentation tank. Okay. Now let's talk about maturation. After the fermentation is done properly, we need to have a maturation stage, because in the maturation stage we are having some important features. What are they? The beer is matured for a period of during which the off flavor components like diacetyl, is assimilated by the yeast cells. Okay, so these off flavor components are kind of get rid of at this particular time. And now the mature beer is then chilled to a temperature of -2 degrees Celsius for a couple of days. Okay, and by doing so, the colloidal stability of the beer is greatly improved. That's a very important point, because maturation, when it's done at temperature -2 degrees Celsius, sorry, let me take a color from here. Okay, let me take a color. Yeah, so maturation is usually done at -2 degrees Celsius temperature, when the colloidal stability is a kind of very much increased at this particular stage. Now when the beer is ready to final stabilization stage, where it is there are some proteinaceous matters or tannins still present there, and that's why we need to filter those this proteinaceous materials and tannin out of our beer, because they can produce some kind of bad taste and odor. That's why we don't require them. So for that reason, the final stage of clarification is done, and then the oxygen uptake is reduced as much as possible, since the oxygen will damage the flavor stability of the beer. So if the oxygen is there, it will destabilize the flavor of the beer. That so we need to take out all of the oxygen from this place, as well as the proteinaceous compounds, and also some off flavor compounds like diacetyl. Okay, now here we can see two different examples. That in this left hand side we can see the colloidally stable beer, and in this case we can see unstable beer. In the unstable beer, you can see presence of lot of proteinaceous and amino acid compounds, which is making it pretty much unstable, because filtration is required. And if we filter it properly, we get this kind of clear beer. Okay, and these are the different components. Due to the presence of them, they convert this, or they kind of make the beer unstable. Now the final thing is the beer quality testing, which is also required, because the finished beer is either bottled or canned or filled into kegs, right, as you can see here. But in both the way, there may be contaminants, like microorganisms can be present there, like other bacteria can be present there. If yeast present there, a small quantity of yeast present there, it's fine. But if it is some other organisms, like other bacteria, which can produce toxic materials outside there, so we need to get rid of all those microorganisms from there. For that reason, we need to go through a fast pasteurization. Either it is a tunnel pasteurization or a flash pasteurization. If it is a keg, we went through this tunnel flash pasteurization, but if it is a bottle, then we go through this tunnel pasteurization. And after this pasteurization event, we make sure that the beer we are taking, it is free and get rid of any other harmful microorganisms. It is containing the pure quality substances, it is containing the aromatic compounds and everything which is required, right? Now, visually the finished beer, which is you can say an ale, and must form a nice foam on the pouring. Now for that reason, this foaming is also important, and for this foaming part, there are proteins or proteinaceous materials that are present in beer, which causes this foam. Now usually protein and lipid also. Now the protein called lipid transfer protein 1, or LTP1, is a major component, which usually is coming from the barley, is causing this kind of foaming, right? So for that reason, we need to get rid of this LTP compounds, or this protein compound, as good as we can, because there are some other foaming compounds. If they present, the foaming is a kind of unhomogeneous, it's a kind of disbalancing. But if those proteins, the proteins which are present there are of a simple type, of some proteins are also there which can present an homogeneous foaming, which is required, which we want, because this foaming sometimes provide us some beauty of the beer. And about the flavor, the main flavor or flavor characteristic of the beer is a bitter taste coming from the hops. We all know that. Now secondary flavor is from the fruit estery flavors, because we get those flavors from the barley and all this. Okay, so whatever the most flavored compound present in very small quantities, very small quantities. Actually, it was observed that 800 different compounds present there as a flavoring materials, but we cannot find each of them, which is because it is predominantly bitter due to the presence of hop additions. Okay, and occasionally the undesirable flavor components may appear, giving the beer in the off flavors, but we need to get rid of all these things to produce the beer. So we need to take care of all those other kind of problems that are being associated with the flavors, with the foaming, and with the colloidal stability. Okay, if we can make everything clear, then our beer will be clear and it will be produced properly. Okay, so that's it, and I hope that's helpful.